In the realm of scientific discovery, some breakthroughs quietly shake the foundations of what we thought we knew. In this case, the tremor is unmistakable. A pair of cancer drugs, Rapamycin and Trametinib, have extended the lifespan of mice by nearly 1/3. Not by genetic modification, not by artificial enhancement, but by tapping into the very systems that regulate how we age. This may be the most important clue yet, that aging, once seen as inevitable, may be more negotiable than we imagined. Welcome to a story that could change the way we view life, health, and mortality. On May 28, 2025, the journal Nature Aging published a collaborative study by the Max Planck Institute for Biology of Aging and University College London. Researchers reported that two clinically approved drugs, Rapamycin and Trametinib, used together in mice, yielded a staggering increase in lifespan of around 30%. But this isn't just a story about longevity. It's about quality of life, disease resistance, and the potential to rethink the very structure of human aging. The study began with an essential decision: treat mice at 6 months of age. That's roughly equivalent to a person in their 40s, well past developmental stages, yet still before the steep physiological decline of old age. Rapamycin on its own was known to increase mouse lifespan by 15 to 20%. While Trametinib showed more modest gains of 5 to 10%. But together their effects weren't just cumulative, they were synergistic. Mice treated with both drugs lived significantly longer than any group before them. Importantly, these weren't years filled with decline. The mice retained muscular strength, agility, and cognitive sharpness well into late age. Their fur remained thick and glossy, their organ tissue healthier, their immune systems more robust. Even when challenged with disease triggers, they resisted degeneration far better than their untreated counterparts. At the cellular level, the effect is no less remarkable. Rapamycin targets a molecular complex called mTOR, the mechanistic target of rapamycin, which acts like a central control board for cellular metabolism, growth, and repair. When mTOR is too active, cells grow uncontrollably and struggle to clean out damaged components. By suppressing mTOR, rapamycin reduces cellular stress and reboots a vital process called autophagy, the body's natural recycling system. Trametinib, on the other hand, works through the MEK-ERK pathway, a signaling cascade deeply involved in cell proliferation, stress response, and survival. By dialing down this pathway, Trametinib tempers overactive cellular behaviors that can lead to cancer, inflammation, and age-related tissue damage. But the true brilliance lies in their combination. Together, these two drugs are just different but converging pathways that control how the body ages. It's like tuning both the accelerator and the brakes of aging metabolism, slowing decline while improving resilience. Molecular analysis revealed even deeper insights. The combination triggered entirely unique gene expression patterns, shifts that neither drug caused on its own. Many of these changes were associated with better DNA repair, reduced inflammation, improved mitochondrial function, and enhanced protein stability. In the brain, researchers saw fewer toxic aggregates, often a hallmark of neurodegenerative diseases like Alzheimer's. In the liver, fat buildup was reduced. In the heart, muscle integrity was preserved. A particularly exciting finding was the suppression of genes linked to chronic inflammation and the presence of senescent cells. Senescent cells, which no longer divide and secrete harmful inflammatory signals, accumulate with age and contribute to virtually every age-related condition, from arthritis to cognitive decline. Their reduction is a major marker of biological rejuvenation. Rapamycin's story began in the 1970s in a soil sample collected from Easter Island. Originally explored as an antifungal, it was soon found to have immunosuppressive properties, eventually leading to its use in organ transplant medicine. But its surprising effects on lifespan began surfacing in early 2000s studies, particularly those at the National Institute on Aging's Interventions Testing Program. Trametinib emerged from the targeted cancer drug revolution of the early 2010s. Designed to block specific molecular pathways in melanoma, it was never intended as an anti-aging agent. But over time, researchers began to notice overlaps between oncogenic and gerontogenic pathways. Cancer and aging, it turns out, are not just neighbors, they're roommates. Both drugs inadvertently became tools for probing the aging process itself. Now comes the critical question: can this approach be translated to humans? The potential is enormous. Both drugs are already used in medical settings, which could streamline clinical trials. But there are hurdles. Rapamycin is known to suppress immune function, which could elevate infection risks, particularly in older adults. Trametinib can cause fatigue, rash, and cardiac issues at therapeutic doses. The key might be intermittent dosing or dose reduction. In fact, some researchers are already exploring pulsed rapamycin regimens, doses taken once a week or once every 2 weeks, that preserve benefits without compromising immunity. Trametinib's toxicities could be minimized with shorter courses, especially when paired with protective compounds like antioxidants. There's also a growing push to test these compounds, not in isolation, but alongside lifestyle modifications, caloric restriction, exercise, and gut microbiome interventions. Such combinations could multiply the effects further. The study was led by Professor Dame Linda Partridge and Dr. Sebastian Grunker, both internationally renowned for their work on the molecular biology of aging. Their group at the Max Planck Institute is one of the leading forces in uncovering the fundamental biology behind lifespan and health span. This project also received major input from researchers at University College London, highlighting the global and interdisciplinary nature of modern longevity science. What unites their approach is a core philosophy that aging is not a monolith. It is a system of failures, metabolic, structural, and regulatory, that begin to accumulate and spiral out of control. Interventions that target even a few key drivers may be enough to slow the entire cascade. Moving forward, researchers hope to begin trials in other animal models, including larger mammals like dogs and possibly non-human primates. There's interest in tracking biomarkers such as epigenetic clocks, telomere length, or inflammation markers to see how well this dual therapy performs in real-world aging contexts. AI-driven models are also being employed to simulate long-term outcomes and predict ideal dosing schedules. More broadly, this success opens a philosophical door. If aging can be slowed, can it eventually be paused or even reversed? And if so, what does that mean for how we live, how we work, how we plan for the future? This discovery doesn't promise immortality, but it does promise something more tangible and perhaps more valuable: the possibility of living longer without living sicker. A longer life where vitality remains, independence is preserved, and diseases of aging arrive later, weaker or not at all. It's a future worth chasing. If 2 cancer drugs can unlock the aging code in mice, what else might already be in our medicine cabinet, waiting to change everything? Hit subscribe, because this is only the beginning of a new era, where aging itself may one day become just another treatable condition.